We started up talking about P53 and its involvement in cell cycles and whatnot, but what we really haven't talked too much about is there are two major types of reproduction. If you think of a cell as an organism, which really all cells are, they're their own single-celled organisms working. Even in multicellular organisms, the cells themselves are working for the whole, but they are independent. At one point, all cells were single-celled organisms. And the fact that cells even learned or evolved in some way to work with one another to form tissues and organisms and multicellular organisms is just an amazing thing. And to imagine that every cell in your body right now is actually working autonomously but for the whole at the same time. And to think about that, when your cells, most of your cells actually cannot live for your entire lifespan. Almost all of the cells in an organism, and even cells living in a single-celled existence, they all divide or give rise to offspring. And for multicellular organisms, the reasons could be to repair damage, to rejuvenate oneself, for growth and anabolism. For single-celled organisms, it could just be to give rise to offspring, all right, which is one of the main reasons for organisms' existence, is to live and to grow and to give rise to offspring. There are two major types of reproduction by which cells can give rise to new cells, and one of them is asexual reproduction. All right. Now, in asexual reproduction, organisms give rise to identical offspring where the DNA is exactly the same. Asexual reproduction does not involve sexual parts. It does not involve male and female. Really, what's happening is one cell is replicating all of its DNA and then splitting into two, and the resulting two are the identical of the first. Asexual reproduction, especially when we're talking about multicellular organisms, only occurs in cells of your body called somatic cells, which are soma means body. So somatic cells are body cells. There's only one type of cell in the human body, and even all multicellular organisms. Two cells that do not undergo this somatic asexual reproduction is the sex cells. And for the males, it's the production of sperm, and for females, it's the production of eggs. And each of these sperm and eggs has half the DNA that it needs. All right. So when the sperm and egg fuse, the resulting zygote, or the resulting cell, now has half the DNA from the male and half the DNA from the female, and now the resulting organisms have one full set of DNA. Whereas for somatic reproduction, the cell itself has all the DNA that it needs, and it replicates that DNA, so it has two forms of that DNA. And then the cell splits into two, and now the resulting cells have one copy of the DNA each, identical. But one important thing to remember is cells don't just randomly go through divisions. There's something called the cell cycle, which is a highly regulated cycle that is regulated by proteins and enzymes in the cell that act as checkpoints, and they act to safeguard the process so that the process doesn't go out of whack. We talked a little bit about cancer cells, and cancer cells have lost this ability to control their cell cycling. All right, the cell cycle has 3 or 4 major phases. We can go into 4 phases, but the cell spends most of its time in, and we're going to say about 90% of its time in this phase called interphase. All right. Now, 90% of the time the cell spends during interphase, it only spends about a quarter of that time, or really about 10% of that time, in a dividing phase, which is called the mitotic phase. All right. So normally, a cell is in G1, and in G1 it's really called the growth and normal process phase, the growth phase, where a cell is growing, it is going about its normal activities at normal metabolic rates. It's functioning as the cell would, as the cell's purpose is to be functioning. The heart cell is in this phase, working to support heart. And skin cells in this phase are growing and working to serve their function as a barrier between skin. And cells in your tongue that are designed to secrete saliva or enzymes to break down food, they're doing that. And then when a cell wants to begin to divide or receives a signal that they should be dividing, a bunch of enzymes are produced which set it into something called the S phase of interphase, which is called the synthesis phase, or the phase where DNA is replicated. All right, and during this phase, all the DNA duplicates. So you need enzymes and proteins and cofactors and coenzymes that need to help the cell copy and make new DNA as fast as it can. All right, and this is a phase at which if it goes wrong, DNA doesn't get replicated properly, then the cell will abort the mission, and the cell could either destroy itself, or it could just not go into the next phase. It could destroy the DNA that it tried to make. But in this phase, the S phase, all the DNA is synthesized. So after the S phase, you now have 2 copies of DNA. Now, in order to copy this DNA, all right, the chromosomes, which are a compact form of DNA, need to unravel into a form called chromatin. All right, and chromatin is a form of DNA where these long strands have unwound. You can't actually replicate your DNA in a very, very compact form. All right, so in this phase, all right, DNA is replicated. Now the cell receives some signals and it enters something called G2, which is the growth 2 phase. All right, the second growth phase is all about growing and dividing things that are necessary for the cell to split. So you can imagine all the things that need to be catabolized in order for a cell to split into 2. Remember, the cell needs double of everything in order for this to happen. So some of this stuff, it begins to get synthesized. The cell obviously needs to grow and produce lots more of phospholipids, or lots more hormones, lots more proteins, all this to support splitting into 2. You can imagine if you wanted to split into 2, all the things that your body would have to make double of in order to be ready to split into 2. All right. So while this is happening, there's a change in the concentration of certain enzymes that are checkpoints that allow for the cell to say, hey, we're ready to go. We've replicated our DNA, we've doubled all the things we need to double, we've also produced all the enzymes and things that we're going to need to enter these phases. We now enter the M phase, or the mitotic phase. All right. And remember, right now we're talking about asexual reproduction in the somatic cell. All right. Mitosis, not meiosis. All right. And in mitosis, we have the cells that are resulting are identical. They have the same DNA, and in the human body, they are somatic cells, or body cells. All right. Every cell in your body that is not a sex cell. All right. It's basically every cell you can imagine. Then there are 4 major phases within the mitotic phase. There's prophase, metaphase, anaphase, and telophase. And remember, every one of these phases is regulated by its own enzymes and checkpoints as well, so that we know that every time we enter one phase into another, we are fully ready, prepared to go into the next phase. All right. Now after telophase, all right, after you have one cell and it divides into 2, these cells are now both in G1 of interphase, where they spend most of their time, all right, growing normally and doing what their normal purposes are. All right. This is a graph showing different types of proteins and enzymes that are involved with the process of mitosis and interphase. All right. As you can see, cyclin D, cyclin E, cyclin A, and cyclin B were named so because, well, they were named after a bicycle, actually, after the researcher who found them didn't really know what they did. But once they found that they actually were cyclic during the process of mitosis and interphase, the name stuck as cyclins. All right. So basically, you can imagine what cyclins do is they appear and disappear. They are made and broken in cycles throughout the process of interphase and mitosis. All right. What these cyclins do is they set off a chain of reactions that tells the cell that it's either ready or not to continue into the next phase. All right. So you can imagine cyclin E is one of its main purposes is to let the cell know when its concentrations get really high, it means everything is okay, all right, and that we are ready to enter the S phase. And as soon as S phase is entered, cyclin E is degraded, all right. And as S phase begins to continue, all right, cyclin A begins to be synthesized, and as it's synthesized, it's setting off a bunch of reactions that are then having an effect on other things like the presence of cyclin B, because cyclin A goes up and then cyclin B goes up proportionally to cyclin A. Cyclin A gets to a peak, and as it starts to drop, all right, cyclin B reaches peak right after that, and then it starts to drop. And if you see during the process of mitosis, cyclin E has dropped. Cyclin A and cyclin B are now being degraded very quickly. All right. So all of these reactions that are going on in the cell are designed to regulate every aspect of the process so that the somatic cell, can divide properly. All right. So we have this process of the cell cycle. It's important to remember that the entire cycle is regulated by proteins and enzymes. All right. I think no, it's Bdellovibrio. Let's see. We're going to end it for now. So we talked about cyclins a little bit and how cyclins are enzymes and protein enzymes that are involved with binding and setting off reactions that allow the cell to enter from one phase to another of interphase, and also are involved with the beginning of mitosis. And all of these enzymes are degraded by the time mitosis begins, and then other enzymes will regulate the process from then on. If we continue to look at cell cycle proteins, one of the most important things to look at is an enzyme called CDK. All right. Now CDKs are enzymes. They're a family or class of enzymes that are called cyclin-dependent kinases. All right. They're called cyclin-dependent kinases. CDKs. I don't want maps, actually. I do want images, though. Okay. I think what this is going to show us is the presence of various CDKs throughout the process. Okay. So maybe this is a good one here. Yeah, here we go. This is a good one. So CDKs are cyclical, just like the cyclins. All right. And actually, cyclins and CDKs act with one another to allow different parts of the cell cycle to occur. So as G1 enters S, specific cyclin-dependent kinases are binding to cyclins, either allowing them to do what they do, or activating them, or they're degrading them into allowing the next cycle to occur. Like the presence of this cyclin D, all right, is necessary for the transition from G1 to S, except it is not necessary for S to actually occur. So it's necessary for G1 to go to S, but it's not necessary for S. So it binds to the CDK, the cyclin-dependent kinase, and it catalyzes reactions that degrade the cyclin so that by the time we enter S phase, we now have a substrate enzyme complex of cyclin E with CDK2, another CDK, another cyclin-dependent kinase. Remember, these cyclin-dependent kinases are setting off phosphorylation reactions that are allowing the cell to enter from one stage to the next. Again, as cyclin E gets degraded, as the cyclin E CDK complex gets degraded, now cyclin A CDK2 binds to cyclin A instead of cyclin E. Now you can imagine there's some sort of conformational change with this protein, But the important thing is that G2, certain levels of cyclin A, CDK2 concentrations are necessary for G2 to occur into M phase. And again, as we get into M phase, the presence of cyclin A CD2 CDK complex 2 needs to degrade in order for the M phase to occur. All right. So if we move to this next one here, we have the cell cycle proteins. What this is just showing us is the CDK complexes. And what these CDKs do is they catalyze kinase reactions, again, phosphorylation reactions, that allow for each step of the cycle to occur. This CDKG1, all right, is actually going to bind with the G1 cyclin, which is the cyclin D, all right. And when that binds, all right, we can now enter the S phase, or its lack of binding, its degradation allows us to enter the S phase. So what keeps us in G2 phase, all right, is that we have an active CDK cyclin B, or not cyclin B, cyclin D complex. All right. As we enter the S phase, all right, we have certain levels of cyclin E, all right, and as we enter G2 phase, we now have different cyclins binding with different things, setting off kinase reactions, whereby the phosphorylation of things, in this case the mitotic CDK, is activating another cyclin that sets us into the M phase. And it's not important for us really to be memorizing what is binding to what. It's really the most important process is to understand that enzymes and binding and enzyme substrate complexes all influence the movement from one part of the cell cycle to another. All right, and this occurs both in mitosis and meiosis. We're going to start off talking about mitosis and the somatic cell division, but it's important to understand that all of these things that regulate going from one phase to another, all right, are all very important, and without them, the cell cycle wouldn't continue.